Okay, so we will discuss all these things in the single. See, biology is a science which deals with the living. Oh, that is Aristotle. He is also known as the founder of biology. Okay, this classification. What is the difference? Yes, plant cell contains cell wall and animal cell doesn't have any cell wall. But in the case of bacteria, it also containing a cell wall. Okay, and the nutrition, the mode of eating. Okay, that. Hello, வணக்கம் பசங்களா. நான் உங்கள் ஆஷா மேம். எப்படி இருக்கீங்க எல்லாரும்? Yes, so today I am here to explain a very important chapter, that is biological classification. So pasangala, we will complete this chapter in a one shot. Okay, this is a very important and a very easy chapter to study. Yes, so when we are starting our class, we can see a mind map. I put a mind map here. So I put all the content that we need to study in this single session in this mind map. So today we will discuss about 2 kingdom classification and 5 kingdom classification. The kingdoms like Monera, Protista, Fungi, Plantae, Animalia. Again, this all kingdoms are further divided into different categories. Okay, and we have to study about viruses, viroid and lichens. Okay, so we will discuss all these things in the single session. Paklama? Yeah. So tell me, what is biology? Again, I am asking you, what is biology? Biology enna? We already learned it in our previous class. What is that? What is it, biology? Biology is a science which deals with the living organism. As I already mentioned, biology, it is the combination of bios plus logos. These two words combined together to form the word biology. So it means the study of living organism. Clear? And Aristotle is known as the founder of biology. Again, the chapter's name is biological classification. So, pasangala, we have to study about what is classification. What do you mean by classification? It is the arrangement of anything into groups. It is the arrangement of anything into groups. Okay. So, arrangement of anything into groups can be considered as classification. And then, what is biological classification? Ma'am, this is a question. We know the answer because biology is something related to science. So, biological classification is nothing but it is the scientific method of arrangement of organisms into groups. Very good, pasangala. It is the scientific method of arrangement of organisms into groups. Okay, and in biology, the first classification is early system of classification. Listen here, it is proposed by Aristotle. So, early system of classification is proposed by Aristotle. His picture is here. Yeah, what a Superman. Yes. So, early system of classification is proposed by Aristotle, and he classified all the animals around us into red-blooded animals and non-red-blooded animals. Okay, see, he classified all the organisms around us into red-blooded animals and non-red-blooded animals. Again, based on morphology, he classified plants into trees, shrubs, herbs. Okay, this is the classification proposed by whom? Proposed by our superhero, that is Aristotle. He is also known as the founder of biology. Okay, this classification is not that much established. So, we need to study about the second classification, that is 2 kingdom classification. So, this classification is proposed by, yes, next superhero. What's his name? Carl Linnaeus. He proposed the second classification, that is 2 kingdom classification. Based on 2 kingdom classification, he classified all organisms around us into 2 main categories, that are kingdom Plantae and kingdom Animalia. So, he classified all the organisms around us into 2 main categories. First one, kingdom Plantae, and the second one, kingdom Animalia. So, from the name itself, it is very clear, pasangala. Ma'am, kingdom Plantae plants irukke, and when we come to Animalia, it has lots and lots of animals. Okay, he classified all the organisms into two major broad categories. But there are some exceptional organisms are there. bacteria, fungi. Where can we include all these organisms? Is bacteria a plant? Is bacteria is an animal? Yeah, we have this type of confusion. So obviously, what two kingdom classification has some drawbacks. We already learned it in our second chapter. We just need to read it. These are the drawbacks. Did not distinguish between eukaryotes and prokaryotes. Pasangala, there are so many organisms. Okay, eukaryotes and prokaryotes. Prokaryotes mean they don't have a well-defined nucleus. The organism which don't have a well-defined nucleus can be considered as prokaryotes, and eukaryotes have well-defined nucleus and cell organelles and everything. Okay, plants, animals are eukaryotes. So this is the first drawback of two kingdom classification. It will not describe or it will not tell anything about the eukaryotes and prokaryotes. Okay? Yeah. Next point is did not distinguish between unicellular and multicellular organism. Pastangala, there are lot of organisms like uni. What do you mean by unicellular? Unicellular. Uni. What is the meaning of uni? Only one cell. So there are some organisms which have only one cell in their body that are called unicellular organisms. Yeast is an example, bacteria is an example, amoeba is an example for unicellular organisms. And there are lot of multicellular organisms are there. Multicellular, multi. What's the meaning of multi, Pasangla? Many. Okay, many. Okay, so we are many cells. Okay. We are not many cells. Yes, it contains multicellular organisms contain many cells. Don't forget this. And so the second drawback is did not distinguish between unicellular and multicellular organism. Did not distinguish between photosynthetic and non-photosynthetic organism. Okay, photosynthetic organism. What are they? Plants, cyanobacteria. These are some photosynthetic organisms. So in two kingdom classification, Linnaeus is not mentioned about what photosynthetic and non-photosynthetic organisms. Large number of organisms did not be considered. So these are the major drawbacks of two kingdom classification. Is that clear, Pasangla? So you should remember two kingdom classification is proposed by Carlus Linnaeus, and based on his classification, he classified all the organisms into two: kingdom Plantae and Animalia. Kingdom Plantae include plants and kingdom Animalia includes animals. Okay, va? Yes, now the doubt Pasangla. If you have any doubt, feel free to comment in the comment box. Okay. So next we have to upgrade the classification, that is five kingdom classification. Okay, and it is proposed by R.H. Whittaker. This scientist. Yeah, wait a second. Yeah, this classification is what happened to my pen? Yeah, I will tell. So, five kingdom classification is proposed by whom? It is proposed by R.H. Whittaker. Five kingdom classification is proposed by R.H. Whittaker. Okay, he chose some criteria for classifying the organism, and these are the criteria: the cell structure, thallus organization, mode of nutrition, reproduction, phylogenetic relationship. Phylogenetic relationship means the evolutionary relationship. So he chose these criteria for classifying the organisms into five kingdoms, and based on this criteria, what are the five kingdoms? Repeat it with me. Yeah, that are Monera, Protista, Fungi, Plantae, Animalia. Ma'am, enna chodyam idu? We can say it in our sleep also. Okay, so what are the five kingdoms? Monera, Protista, Fungi, Plantae and Animalia. And it has some advantages, Pasangla. First advantages, clearly separate prokaryotes and eukaryotes. Okay, so it gives a clear idea about what prokaryotes and eukaryotes. It gives a clear idea about prokaryotes and eukaryotes based on scientific features, based on, wait a second, I will underline it, otherwise you guys will not notice this. Okay, so based on scientific features like cell type and nutrition. So this is the second advantage. So here R.H. Whittaker considered the cell type and nutrition, the mode of eating in it. Okay, and gives fungi and protista their own kingdom. This is also very important. Fungus has its own kingdom and protista has its own kingdom. Yes, shows evolutionary and ecological relationship among organisms. So it considered the evolutionary and ecological relationships also. So these are the major 5 kingdoms. So we have to study about each and every kingdom. First of all, Kingdom Munira. Kingdom Munira. What does this image indicate? Who is this? இது சொல்ல முடியுமா? என்ன இது? Yes, this is bacteria. This is the picture of what? This is the picture of bacteria. So, bacteria are the sole members of Kingdom Munira. Bacteria are the, wait a second, yeah, bacteria are the sole members. These are the sole members. Bacteria are the sole members. Okay, these are the sole members of Kingdom Munira. And Kingdom Munirans are unicellular. As I already mentioned, bacteria is a single-celled organism. So, it is unicellular. Prokaryotic. Prokaryotic means it doesn't have a well-defined nucleus. No well-defined nucleus. No well-defined nucleus. No well-defined nucleus. It is a speciality of Munirans. Okay. So, Muniras are known as prokaryotic and it is microscopic. We need to use a microscope for viewing this Munirans. Okay, so you should remember Munira mainly bacteria is a sole member. It is unicellular, prokaryotic and microscopic. Lack definite nucleus and membrane bound organelles. That's why they are called what? Prokaryotes. Rigid cell wall is present. One thing is very important. It is unicellular, microscopic and prokaryotic. But it has what? It has a rigid cell wall. It has a rigid cell wall. What is the difference between plant and animal cells, Pasangala? What is the difference? Yes, plant cell contains cell wall and animal cell doesn't have any cell wall. But in the case of bacteria, it also containing a cell wall. Okay, and the nutrition, the mode of eating. Okay, that is heterotrophic and autotrophic. Heterotrophic means it will depend on the organism for eating their food or for their food. And autotrophic means it can synthesize their own food in their own body. Okay, so some of the bacteria are what? Autotrophic and some of the bacteria are heterotrophic. You should remember these points also. Yes, again, look into this picture, Pasangala. Here I put a flow chart. Yeah. Here you can see that Kingdom Munira is further classified into different categories that are Archaebacteria, Eubacteria and Mycoplasma. Okay, Archaebacteria, repeat it with me, Archaebacteria, Eubacteria and Mycoplasma. Archaebacteria again is of 3 types. Ma'am, what is this? There's so many classification. Confusion. Don't worry, I will clear these confusions in this entire session. You should follow my class. Okay, so Archaebacteria, again we need to, this is an ancient kind of bacteria. It is a primitive, ancient or primitive bacteria. And again, I told you it is classified into halophiles. Halophiles means the archaebacteria which love salt. Salt-loving bacteria. Some bacteria are loving salt. They need salt to live. Okay, so halophiles, salt-loving bacteria are here. Thermoacidophiles. Thermo. What do you mean by thermo? I think you guys already learned this thermo word in physics, thermodynamics. Yes, what is thermo? Ma'am, thermo is something which is connected to temperature. Yes, that's correct. So, thermoacido. It is you learned in chemistry. So, biology here you can see the combination of both chemistry and physics. Okay, so thermoacidophiles, it is the bacteria which live in hot springs and it is acid loving. It is acid loving bacteria. It is acid. They are acid loving bacteria. And one more category is there, that is methanogen. What is that? Methanogen. So it is present in the marshy areas, the dump areas or the ruminant animals' gut. Okay, what are ruminant animals? The grass-eating animals like cow, buffalo, these are ruminant animals. So, this methanogen bacteria is present in where? It is present in marshy areas, ruminant animals' gut. Okay, and why it is called methanogen? Because it's produce methane. Okay, produces what? It produces methane. So in kingdom Onera, there is a kind of bacteria that is called Archaebacteria. It is a primitive or ancient kind of bacteria. Again, it is classified into halophile, thermosidophile, and methanogen. Halophiles, salt-loving; thermosidophiles, acid-loving and lives in hot spring. Methanogen, methane-producing bacteria and lives in marshy areas and gut of ruminant animals like cow and buffalo. Yes, next we have to go to the next category, that is eubacteria. Kingdom Monera is further classified into Archaebacteria, eubacteria, mycoplasma. Guys, please repeat it with me, otherwise you can't memorize all these things because these terms are very new to you, so you have to repeat it with me. So let us have a look into eubacteria. These are known as the true bacteria. So these are known as the true bacteria. Again, it is further classified into bacteria and cyanobacteria. So eubacteria further classified into what? Bacteria and cyanobacteria. Bacteria is of 4 types, singular. Based on the shape of the bacteria, it is classified into 4 types, that are Coccus, Bacillus, Vibrio and Spirillum. These are the 4 major kinds of bacteria based on the shape. Okay, look into this picture. Coccus. What is the shape of coccus? Ma'am, this is spherical shape. Okay, so coccus are spherically shaped bacteria. Okay, it may be occur in a group, or it can be seen as a single one, it can be seen as in groups. Okay, so it may occur in singly, that one of the example is monococcus. Okay, and in pairs, diplococcus. If it is in pairs, it is called diplococcus, and if it is in groups, staphylococcus. This is another example. Or we can see it as a long chain, that is called streptococcus. So bacteria which have a what? Spherical shape is called coccus. Okay. Yes. Next one is bacillus. Yes, we can see that in bacillus it has a rod shape. Okay, it has a rod shape, and it is also occurs in singly, pairs, or in groups. Okay, yes, singly that is called bacillus, and in pairs it is called diplobacillus, and or in groups streptobacillus. Okay, and the next one, the third one is Vibrio. Yeah, Vibrio. What is the shape of this? What is the shape of this? Yeah, ma'am. This is comma allava. Ade makkale, this is comma. So here we can see a comma. Okay. So Vibrium are basically comma shaped bacteria. Okay. One of the examples is Vibrio comma. Yes. The next one is Spirilla. What is that? Spirilla. Next one is Spirilla. Yes, it can be seen here. It has a spiral shape, and the major example is Spirochetus. So you should study this singular. Bacteria is of 2 types, bacteria and cyanobacteria. Yes, bacteria can be again classified based on their shapes, that are Coccus, Bacillus, Vibrio and Spirillum. Okay, va? Yes. Next, the mode of nutrition of bacteria. I already told you bacteria are of 2 types, autotrophic bacteria or heterotrophic bacteria based on their nutrition. Autotrophic bacteria can be further classified into 2, that are photosynthetic autotrophic and chemosynthetic autotrophic. From the heading itself, it is very clear. Photosynthetic, what do you mean by photo? Singular? Photo means light. Photo means what? Photo means light. And what do you mean by chemo? Chemical. Okay, let us read this. Photosynthetic autotrophs. They have chlorophyll A. This is very important. It has chlorophyll A, similar to that of green plants. Some of the bacteria already containing chlorophyll A in their body, like green plants. Okay, cyanobacteria is one of the major example for it. What is the other name of cyanobacteria? Please comment in the comment box. What is the other name of cyanobacteria? What is that? Please comment it. I will tell this in the next class. I will read the comments in the next class. Okay, please comment it. Yes. So, photosynthetic autotrophs, these are the bacteria which has the ability to produce their own food in their own body. Okay, because it has chlorophyll A in its body. Next, chemosynthetic autotrophs. Chemical, they oxidize inorganic substances such as nitrates, nitrites and ammonia. Okay, and they used to release energy for their ATP production. So this is the process, this chemical process is happening inside this bacteria's body. Sulfur bacteria, sulfur bacteria is one of the example for this. Sulfur bacteria. Yes, sulfur bacteria is one of the example for this. Okay, and they play a great role in recycling nutrients. They play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur. Okay, this is the first mode of nutrition, that is autotrophic mode of nutrition. The second one, heterotrophs. I told you some of the bacteria are heterotrophs. They depend on the organism for their food. Most abundant in nature. It is most abundant in nature. Majority are important decomposers. What do you mean by decomposers, Pasangala? Decomposers are nothing but the bacteria which will feed on dead and decaying matter. Okay, yes, they are used to make curd from milk. This is also a benefit of this bacteria. It can be used for making curd from milk. Okay, which bacteria present in milk? Which bacteria is present in milk, or which bacteria can be used for converting milk into curd? That is lactobacillus. Okay. Then what is the shape of lactobacillus? I'm repeating, bacillus. Ma'am, it has a round shape. Okay, that's correct. Yeah. Yes, heterotrophs. They are used to make curd from milk and production of antibiotics. Okay, production of antibiotics. This is also an added advantage of what? This bacteria. Yes. Then next, it will fixing nitrogen in legume roots. Legume roots. So bacteria, the heterotrophs are of different types like saprophytes, parasites. Then what is the last one? Saprophytes. Parasites, please comment in the comment box. Parasites are the organism which will depend the host organism for their food. Saprophytes will feed dead and decaying matter. Okay. And the third one I will tell you, that is symbiotic bacteria. Okay, nitrogen fixing bacteria is an example for symbiotic bacteria. Symbiotic means the mutual beneficial relationship. The bacteria which provide or which will have a mutual beneficial relationship with other organism. Okay, here in the case of legumes, rhizobium is the bacteria. Okay, rhizobium is the bacteria which is making a symbiotic relationship with what? With leguminous plants. Okay, and some bacteria are pathogens, Pasangala. It will cause diseases. In human beings, it will cause us so many diseases, and in the case of plants, it will cause us so many diseases. Okay. So next we have to discuss about the reproduction in bacteria. This is also very important. Reproduction. What is reproduction? Enna chodhyam idu ma'am? We know the answer. Okay, in the ma'am vandha niraya chodhyams you are asking, but we know those answers. I know, Pasangala, you were very great. My students are very great. You know the answers. That's why I am asking again and again. Yes. So what is reproduction? It is the production of next generation. So bacteria reproduce mainly by fission. This is the process. Main process occurs in bacteria's body, that is fission. What is that? Fission. And under unfavorable condition, they produce a spores. So under unfavorable conditions, they will produce a spores also. And they also reproduce by sexual reproduction. So bacteria has different kinds of reproduction. First of all, it will reproduce through binary fission. Binary. What do you mean by bi? Bi means two. So what is binary fission? One bacteria is divided into two. This is a picture of binary fission. This is a picture of binary fission. and some under unfavorable condition, it will produce as what? It will produce as spores, and also bacteria has the ability to reproduce through sexually. Okay, that is called conjugation. That kind of sexual reproduction is called conjugation. You should study the name conjugation. Conjugation. In conjugation, they will transfer the DNA. They will transfer the DNA from one bacterium to another. Okay, so you should remember this also. This is the mode of reproduction in bacteria. Next, we have to study about the cyanobacteria, pasangala. Cyanobacteria. Cyanobacteria. I already told you to comment in the comment box what is the next or other name of cyanobacteria. I will tell you that is blue-green algae. Blue-green algae. Blue-green algae. When we hear this term, we will just think, is cyanobacteria is an algae? Is that an algae? No, Pasangala. It is not an algae. It is a bacteria. Okay, cyanobacteria is single-celled. Single-celled. The example is spirulina and can be seen in colonies. Okay, colonial. That is called a nostoc. The example is nostoc. And filamentous cyanobacteria are there. Oscillatoria is one of the example for it. Oscillatoria. Colonial forms of cyanobacteria are generally surrounded by a mucilaginous sheath. So, look into this picture too. Okay, here this picture we can see the mucilaginous covering, the outer mucilaginous, the blah covering, not covering him, the covering. Okay, this is called mucilaginous sheath. Okay, so colonial forms of cyanobacteria have what? A mucilaginous covering, and it prevent. What is the function of this mucilaginous sheath? It will prevent drying of the cells during desiccation. One of the major example is nostoc. Nostoc is one of the major example for it. Okay, and as we already learned, cyanobacteria containing which pigment? Yes, chlorophyll A is the pigment present in cyanobacteria. Maybe cyanobacteria can be seen in aquatic areas, or sometimes we can see in terrestrial areas. They form blooms in polluted water bodies. So, cyanobacteria, pasangala, this is an indication for pollution. It is an indication for pollution. They form blooms in polluted water bodies. Okay. Some of the cyanobacteria will fix nitrogen. What? Will fix atmospheric nitrogen. And there is a specialized cell. Here we can see heterocyst. Heterocyst, this violet colored structure. Okay, this violet colored structure in the second picture, that is called heterocyst. This heterocyst is in cyanobacteria is for fixing atmospheric nitrogen. It is for fixing what? It is for fixing atmospheric nitrogen. You should remember these points. Yes, next category. That is mycoplasma. What do you mean by mycoplasma? What is that? Mycoplasma. Pasangala, this is the very smallest living thing in the world. Okay, they are what? Wait a second, I will show you. Yeah, I told you Monera is of three types. Archaebacteria, eubacteria, mycoplasma. That mycoplasma we need to study now. Mycoplasma is a very tiny structure. It doesn't have any cell wall. The organism without a cell wall. These are the organisms without a cell wall. They are the smallest living cells, not. They are the smallest living cells, not. They can survive without oxygen. This is a very important thing. They can survive without oxygen. So, mycoplasma can survive without oxygen. ஏதாவது doubt இருக்கா பசங்களா? ஓகேவா? Continue பண்ணலாமா? Yes. So, mycoplasma can survive without oxygen. Many are pathogenic in animals and plants. What do you mean by pathogenic? The disease causing agents are called pathogens. So, many of the mycoplasmas are what? Pathogenic. The disease causing agents. Okay. Yes, this is a picture of mycoplasma. Clear? Clear? Very good. So, let us move to the next, the second kingdom, that is kingdom Protista. Okay. What is that? Kingdom Protista. It includes single cell eukaryotes. So, in Protista, we can see single cell eukaryotes. In monerans, prokaryotic organisms are present in monerans. Okay. When we come to Protista, it changed. Okay, eukaryotes are present in where? Eukaryotes are present in kingdom Protista. It contains eukaryotes. Protistas are primarily aquatic. Mostly these protistas are what? They are aquatic. They are only present in water. They are only present in water. It forms a link with plants, animals and fungi. It has a connection with plants, animals and fungi. Cells contain well-defined nucleus and their membrane-bound organelles. So, as I mentioned, it is a eukaryote. It is a eukaryote because it has a well-defined nucleus and well-defined nucleus and membrane-bound organelles are present. And some of the protista containing flagella, flagella or cilia. Do you know what is flagella? Yeah, let us take this picture. This is the picture of Euglena. This is a picture of Euglena. It has this tail-like structure. Okay, this is called flagella. This tail is called flagella. This is called flagella. Pasangala, this is for their locomotion. So flagella and cilia are the two structures present in protista for their movement. We have legs to move, right? Yeah, like that. This protista have flagella and cilia for their movement of the locomotion. And it can reproduce asexually, sexually by a process involving in cell fusion, zygote formation. Okay, these are some pictures, some examples of protista. Amoeba, amoeba, euglena, slime mold, diatom, paramecium, dinoflagellate. These are the major examples of protista. Again, these protistas are of 5 types. Pasangala, as we already discussed our chapter's name, biological classification. So in this entire session, we have to study more and more, many classifications. Okay. So protistas again classified into chrysophytes, dinoflagellate, euglenoid, slime molds and protozoans. I will explain this 5 in the next slide. Chrysophytes. What are chrysophytes? It's a kind of protista. Chrysophytes is a kind of protista. They are found in fresh water. Okay, it is found only in where? It is found only in fresh water. Okay, so let us have a look into chrysophytes. Chrysophytes found in freshwater and marine environment. Where can we see this? It is present in where? It is present in freshwater and in marine environment. They are microscopic. So chrysophytes are microscopic, float passively in water currents. Okay, plankton is one of the example for it. So, chrysophytes are freshwater. It is present in freshwater and in marine environment. It is microscopic. We need to use a microscope for viewing this chrysophytes. Okay, and they float passively with the water current. They can flow passively with the water current. Most of them are photosynthetic. Pasangala, chrysophytes. Most of the chrysophytes are what? Photosynthetics because of the presence of chlorophyll in it. It includes diatoms, golden algae, that is desmids. Okay, desmids. So these are the major important examples for chrysophytes. Okay, diatoms. What is diatom? Diatoms have silicaceous cell walls that form 2 overlapping shells. That is called diatom. That is why it is called diatom. The cell wall deposit of diatoms form diatomaceous earth. Pasangla, yes, diatoms contain silica walls. Okay, and the disposition of the silica wall at the bottom of the ocean makes the ocean as the diatomaceous earth, a silica deposition at the bottom of the ocean. Yes, that is called the diatomaceous earth, which is used for polishing and filtration. It has some economical importance that are polishing and filtration. Polishing and filtration. Diatoms are the chief producers. Who are producers? I am not asking about the film producers. I am asking about the chief producers. Okay. Producers are the organism which has the ability to produce their own food. They are in their body. Okay, yes, that are called producers. Plants are mainly known as the producers in land. When it comes to ocean, chief producers are diatoms. Chief producers are diatoms. This is the picture of diatoms, Vasangla. This is the picture of diatoms. Next one is dinoflagellates. Next one is dinoflagellate. Mostly these are marine and photosynthetic. This is also marine and photosynthetic. They appear in different colors. Yellow colors are there, green color is there, brown, blue, red, so many colors are there based on the pigments present in it. Based on the pigment present in it, dinoflagellates are in different colors. Okay, and its cell wall has a cellulose plate. Its cell wall has a cellulose plate. Most of them have two flagella. Most of them have two flagella, Pasangala. One is longitudinally and the other one is transversely. Okay, yes, and one of the major example of this dinoflagella flagellate is goniolax. Goniolax. Red dinoflagellates. They are known as the red dinoflagellates because it is present in red color. The pigment present in it is red, so it is present in a red color. The name is goniolax. It is very, very important because it undergoes rapid multiplication so that it will increases its number and appears as a red color. So it can the ability to make red tides. Okay, so goniolax, that is red dinoflagellate, which has the ability to produce red tides. Okay, and it also releases some toxins, Vasangala. It also releases some toxic substances, toxins, which is harmful to marine animals such as fishes and other organisms. You should remember this. This is the picture of dinoflagellates. It is also a part of Protista. The next one is the third category is euglenoids. What is that? Euglenoids. They are mainly freshwater organisms. Okay, chrysophytes and dinoflagellate both are present in aquatic and marine region. But when it comes to euglenoids, it is only present in where? It is only present in fresh water, and it can be found in stagnant water. Stagnant water means കെട്ടിക്കിടക്കുന്ന വെള്ളം. Okay, കെട്ടിക്കിടക്കുന്ന വെള്ളം. It is present in stagnant water. Instead of cell wall, they have a protein-rich layer, Pasangala. It doesn't have any cell wall. Instead of cell wall, there is a protein-rich layer that is called pellicle. What is that? Pellicle. There is a protein-rich layer present in euglenoid that is called pellicle. You should remember the name pellicle. It makes their body flexible. So this pellicle made its body flexible. They have two flagella. It also euglenoids also containing two flagella. One flagella is very short and the other one is very long. Okay, one flagella is very short and the other flagella. What is the function of flagella? Comment in the comment box. Flagella is for their locomotion, for their movement. So, one flagella is short and the other one is long. They are photosynthetic in the presence of sunlight. So, when it gets sunlight, it has ability to do or perform photosynthesis. Or in the absence of sunlight, most of them are heterotrophs. They will predating on other small organisms. They are becoming predators when they didn't get any sunlight. Okay, va? Yes, the pigments of euglenoids are identical those of higher plants. One of the major example is Euglena, Pasangala. Euglena is one of the major example. This is the picture of what Euglena. Here you can see the large flagella. Clear? Without a doubt? All clear? All good? Yes. So let's move to the next one, that is slime mold. Slime mold. These are saprophytic. What do you mean by saprophytic? They will feed on dead and decaying matter. These are saprophytic. They will feed on dead and decaying matter. Decaying matter. They will feed on dead and decaying matter. Okay. The body moves along decaying twigs and leaves, engulfing organic material. Okay. It has the ability to engulf the organic material, dead and decaying organic material. Under suitable conditions, they form an aggregation called plasmodium. Okay, we already learned another plasmodium, that is the causative agent of malaria disease. Okay, I am not talking about that plasmodium. This is another plasmodium. This is the aggregation, the group of what? Slime mold. Under suitable condition, this slime mold will make an aggregation that is called plasmodium. This is not the plasmodium which causes malaria. Okay. Yes. During unfavorable conditions, what will happen? The plasmodium differentiate and forms fruiting bodies bear spores. Okay, so during unfavorable condition, it will make some spore. It will make some spores during unfavorable, during unfavorable conditions. Okay. Yeah, the spores are very strong and it contains true walls. Okay, they are extremely resistant and survive for many years. Okay, and the spores dispersed by air currents. Air currents can be easily dispersed through air. Okay, so this is about slime mold. This is the picture of slime mold. Next, we have to discuss about the last protista, that is protozoan. Protozoan. They are heterotrophs. They are heterotrophs like predators and parasites. Predators and parasites. They are believed to be primitive relatives of animals. It has a close relationship with whom? With animals, and it is again classified, Pasangala. It is of different types, 4 types, that are amoeboid protozoa, flagellated protozoa, ciliated protozoa, and sporozoan. Yes, amoeboid protozoan. It is live in fresh water, seawater, or moist soil. They move and capture prey by putting out pseudopodia. Amoeba. What is the speciality of amoeba? It is pseudopodia. False feed is there. Yes, amoeba is one of the major example. Marine forms have silica shells on their surface. Some are parasites. Endamoeba is another example for amoeboid protozoan. Endamoeba is a pathogen also. Okay, it has the ability to create some diseases. Amoebiasis, that is the name of the disease. Yes, next one is flagellated protozoan. From the name itself, it is very clear. It has flagella. It is free living. They have flagella. Parasitic forms cause diseases like sleeping sickness. Sleeping sickness is a disease caused by this flagellated protozoan, and this disease's name is trypanosoma. Trypanosoma. Next one is ciliated protozoa. Ciliated. It has cilia. Okay. Based on the structure present in it, it is classified into this. Okay, so amoeboid protozoa, flagellated protozoa, ciliated protozoa, sporozoan. Next one, sporozoa. It is infectious. It is infectious. It causes diseases. Here comes the other plasmodium. The plasmodium which causes malaria is present under sporozoan. These are the images of this protozoans, that are endamoeba, trypanosoma, paramecium, plasmodium. Okay, clear? Okay, va? Yes. Next, we have to move to the third kingdom, that is kingdom fungi. Kingdom fungi. It is a unique kingdom of heterotrophic organism. Ajangala, this kingdom only eat their food through heterotrophic nutrition. They don't have any autotrophic kind of nutrition. These are heterotrophic organisms. Most of them are saprophytic. Saprophytic means they will eat dead and decaying matter. Fungi are cosmopolitan and occur in air, water, soil. It is present in everywhere, Pasangla. This fungi is present in everywhere. They prefer to grow in warm and humid places. Where can we see this? It is present in warm and humid places. It needs a moisture content to grow. Fungi needs a moisture content to grow. Most of the examples are bread mold, orange root, mushroom. These are the examples. Ma'am, mushroom. I think mushroom is good to eat. Is that good to eat? Is fungi is good to eat? Correct, Pasangla. Some of the fungi are edible. Mushroom is the best example for it. Okay. Not all fungi are edible. Some of the fungi are edible. Like yeast is also a fungus. Yeast is also a fungi. Yeast can be eat. Mushroom can be eat. Okay, so white spots on mustard leaves are due to parasitic fungus. This is also one another example. Some fungi are the source of antibiotics, Pasangla. Penicillium. Penicillium is the example for it. You know, penicillin, the antibiotic penicillin. This is the first discovered antibiotic. It is from a fungus, Penicillium. It is discovered by Alexander Fleming. Yes, some fungi are unicellular. Okay, example is yeast and the other are multicellular. Yeast can be used for making bread, beer. Okay, yeast can be used for making bread and beer. Other fungi causes diseases in plants and animals. Wheat. Okay, Puccinia causes rust. Okay, wheat in rust. This is also a disease caused by the fungus Puccinia. Pasangla, here you can see so many pictures. These are the pictures of what? These are the pictures of different fungi. This is the picture of bread mold. Puccinia is here. This one. Yeah. This one is the Puccinia. Penicillium is here. The fungi that we can use for making antibiotic penicillin. Yeast is here. Okay. Yes, these are the examples, Pasangla. Clear? Anybody have any doubt? Don't worry, just comment in the comment box. I will reply it in our live or in the next class. Yes, except yeast, fungi are filamentous. Okay, except one fungi, that is yeast. Other fungi are what? Filamentous in nature. Filamentous in nature. Their body consists of long, slender, thread-like structure called hyphae. So, Pasangla, fungal body is made up of hyphae. Fungal body is made up of what? Fungal body is made up of a structure called hyphae. The filamentous, long, slender, thread-like structure called hyphae. And the network of hyphae is called mycelium. The network of hyphae is called mycelium. This is the network of hyphae. Okay, mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. Some of the hyphae containing what? Many nucleus, many nucleus in a single hyphae that are called choanocytic hyphae. Choanocytic. Some of the peoples are pronouncing like sinusotic hyphae. That is also correct. You can pronounce whatever comes into your mouth. Okay, sinusotic or choanocytic, both are correct. Okay, so this is the speciality of some of the fungus. Others have a septate or cross walls in hyphae. Look into this pictures, Pasangala. Here you can see the picture of hyphae. This is the picture of hyphae. This one is the picture of hyphae. So you can see the, wait a second, I will mark it. Yes. So in this hyphae, this is called, the single one is called hyphae and the group of those hyphae are called mycelium. And when it comes to this part, yeah, some of the hyphae containing cross walls in between the nuclei that are called septate hyphae. And some of the hyphae didn't have any cross walls in between them. That kind of hyphae are called choanocytic or xenocytic hyphae. Clear? Yes. Next, the mode of nutrition of fungus. What is the mode of nutrition? I already told you. They are heterotrophs and the major mode is saprophyte. Major mode of nutrition is saprophytic. Okay, so they are called, most of them are called saprophytes. They absorb the organic matter, the dead and decaying organic matter, and some of them are parasitic. Some of the fungi are parasitic and some of them are symbionts also. Symbionts means the mutually beneficial relationship that I already mentioned in the case of bacteria. Yes. And the reproduction. Reproduction occurs in 3 types in fungi, that are vegetative mode of reproduction, asexual mode of reproduction, sexual mode of reproduction. What are they? Vegetative, asexual and sexual. Vegetative means fragmentation. It is done by fragmentation process, fission and budding. Okay, and when it comes to asexual mode of reproduction, a specialized structure is formed, conidia. Okay, conidia. A specialized structure is formed, conidia. Conidia, specialized structure is formed, and by sporangiospore, and by zoospore. These are the different mode of asexual reproduction. And in the case of sexual reproduction, it is occurs with the help of plasmogamy, karyogamy and meiosis. With the help of plasmogamy, karyogamy and meiosis. Plasmogamy, pasangala, it is the fusion of protoplasm, and karyogamy is a fusion of nucleus, and meiosis, it is the division. It is a division occurs. I will explain it with the help of this picture. Yeah, here in this picture you can see, look at this picture. Here you can see 2 hyphae. 2 hyphae, mating hyphae. This is the first hyphae and this one is the second hyphae. Okay, these hyphae are combined together to form mycelial hyphae. Again, this combination, the combination of protoplasm is occurs, so dikaryotic stages occurs. I already told you protoplasm will fuse in plasmogamy. So here after plasmogamy we can see 2 nucleus. Look into this picture. You can see 2 nucleus. Blue nucleus is here and red nucleus is here. Blue, red. Blue, red. Okay. So this phase is called dikaryotic stage, or this hyphae is called dikaryon. Okay, dikaryon. 2 nuclei are present in it. Wait a second. What happens to my, is it not? Wait a second, Basangla. I will explain. Yeah, so it contains 2 nuclei in a single hyphae. Okay, this kind of hyphae are called dikaryotic hyphae. Dikaryotic hyphae. Clear? Yes, this is a mode of sexual reproduction, and in it we can see that again the karyogamy, the fertilization is occurs. The fusion of these 2 gametes is occurs. Again, meiosis, the process meiosis is occurs by the after the meiosis. Here we can see spores are formed. Spores are formed. 4 spores will formed after the meiosis. So I will explain it again, Pasangula. Wait a second. Yeah. So in sexual reproduction, first of all, first step in sexual reproduction is plasmogamy. After plasmogamy, karyogamy will occur. After karyogamy, meiosis will occur. Due to the meiosis, spores will produce. The spores will germinate to form the hyphae again. Again, this hyphae will fuse to happen. Plasmogamy, karyogamy, this is like a cycle. The sexual reproduction is like a cycle in fungus. Clear? Clear? So reproduction is of 3 types: vegetative, asexual and sexual reproduction. Yes. Next, fungi is of different types, Pasangala, that are phycomycetes, ascomycetes, basidiomycetes and deuteromycetes. 4 types of fungi is there. 4 types of fungi is there. We have to study the characteristics of each and every fungi. Okay, first one is phycomycetes, that is also called lower fungi. What is that? Lower fungi. It is found in aquatic habitat. Where can we see this? It is found in aquatic habitat, decaying wood and damp places. These are obligate. Obligate means these are the complete parasite. These will depend the host. These will completely destroy the host. Okay, this will completely destroy the host by acquiring nutrients and all the other important parts from it. Okay, so phycomycetes are already known as lower fungi. They will feed on what? Feed on the host organism. Okay, they completely feed on the host organism. So it is called the obligate parasite. Mycelium is aseptate. Aseptate. There is no septa present in where phycomycetes, mycelium, and sinusoidea. So more than one nucleus is present in it. Asexual reproduction by zoospore. Zoospore means the motile, the spores which has the ability to move. That spores are called zoospore. So they are motile. And aplanospore, the non-motile spores are also present in phycomycetes. So asexual mode of reproduction is by zoospore and non-motile aplanospore. And spores produce endogenously in sporangium. There is a structure called sporangium. Sporangium. This kind of structure is present in it. This is the, wait a second, yeah. This is the image of sporangium basangula. This is the image of sporangium. Okay. And the spores are present inside the sporangium so that it is called endogenously. The spores are produced endogenously. Sexual reproduction by zygospore. This is also another kind of spore that is called zygospore. These are formed by the fusion of two gametes. Okay. These gametes are isogamous. Isogamous means sometimes the gametes are similar. Okay, sometimes the gametes are similar. Similar gametes are called isogametes. Or anisogamous are also present. Aniso means dissimilar. Dissimilar or oogamous. Oogamous means female gamete is large and the male gamete is very small. One of the gamete is large and the other is very small. That is called oogamous. Most of the cases, female gamete is large and the male gamete is very small. In the case of human beings, we are oogamous because female gamete, the egg is very large and the sperm is very small. Sperm is very small. You should remember these points. Okay, the examples for phycomycetes are Rhizopus, Mucor, Albugo. Albugo. These are the examples. Yes, Pasangala. Next one is Ascomycetes. Ascomycetes is already known as sac fungi. It is also known as sac fungi. It is unicellular. Example, yeast and Saccharomyces. Okay, yeast is the example. Other one is multicellular. Penicillium is the example for it. So, what are the major important examples of Ascomycetes? Yeast, Penicillium. Clear? Yes, mycelium is branched. Its mycelium is branched and septate. So cross walls are, if consider this is a hyphae of the septas are present in between it. Septas are present in between it. Okay, so mycelium is branched. Mycelium is branched. Mycelium is branched and septate. Cross walls are present in between the nucleus. Okay, and the asexual mode of one more thing. They are saprophytic. They are saprophytic. Decomposers, parasitic. They are saprophytic, decomposers, and parasitic. The asexual mode of reproduction in Ascomycetes by conidia. Pasangala, there is a structure that is called conidia which will exogenously. Exogenously means it will produce spores in the outside of their body. Okay, so exogenously they will produce conidiospore in an extra or a specialized structure that is called conidiophore. This is the structure of conidiophore, Pasangala. This is the structure of conidiophore and these are conidiospores. This is the structure of conidiophore and these are conidiospore. Conidiospore. This is conidiophore. Clear? Yes. Conidia germinate to produce mycelium. Conidia will germinate to produce mycelium. And sexual reproduction is done by ascospore. Ascospore. It is produced endogenously. It is produced endogenously in a sac-like structure, and that sac-like structure is called asci. You can connect it with that song, Vijayana's song. That song. Yes. The song from the film Nanban. Okay, so ascospore, this is produced inside a structure called asci or ascus. Yes, so asci are arranged to form ascocarp, the fruiting body. These are the examples: aspergillus, claviceps, neurospora. These are the major examples. Okay, neurospora is used in biochemical and genetic works also. This is very economically important fungi. Okay, so ascomycetes is commonly called sac fungi. It is unicellular or multicellular. They are mostly saprophytic, decomposers, and parasitic. The hyphae is septate. Okay, because it is it has cross walls in between the nucleus, and it is branched, and the sexual reproduction by producing conidiospore, and the sexual reproduction is by producing ascospore inside a structure called asci or ascus. Next one is basidiomycetes. It includes mushrooms. So the favorite food, mushrooms are present in basidiomycetes. It is also known as the bracket fungi. Or puffballs. Yeah, they grow in soil. It can grow in soil, on logs and tree stumps, and living in plant body also. This is very important. The mycelium of basidiomycetes is branched and septate like the ascomycetes. Okay, the asexual spores are generally not found. Asexual spores are not present in where? Asexual spores are generally not found in basidiomycetes, but vegetative reproduction is happened in basidiomycetes by fragmentation. It is very common. Sex organs are absent in it. Sex organs are absent in it, but plasmogamy occurs by fusion of two vegetative or somatic cells of different strains or genotypes. Okay, yes. It gives rise to basidium. Okay, the resultant structure is dikaryotic structure or dikaryon. So after plasmogamy, a dikaryotic structure will be formed, and it gives rise to basidium. Karyogamy and meiosis occurs in basidium, producing 4 basidiospores also. Basidia are arranged in fruiting bodies called basidiocarp. Okay, examples are agaricus, astilgo, paxinia. These are the examples. Agaricus, mushrooms, smut, and rust fungus. These are the major examples of basidiomycetes. Next one is deuteromycetes. This is also very important, Pasangala. Next one is deuteromycetes. They are commonly known as imperfect fungi. They are not perfect. They are not Mr. Perfect. Okay, so these are imperfect fungi. Not Mr. Perfect. These are imperfect. Mr. Imperfect. You should learn that song like that. Yeah, deuteromites are imperfect fungi because only their asexual or vegetative phase are known. Okay, no sexual reproductive phase is known in deuteromycetes. Okay, deuteromycetes reproduce only by asexual spores, by conidia, by producing conidiospore. Mycelium is again septate. cross walls are present and branch. Some are saprophytes, some are saprophytes or parasites. Some are saprophytes or parasites. Majority are decomposers. Majority of the deuteromycetes are what? Decomposers of litter and help in mineral recycling. Alternaria, these are the example. Trichoderma is the most important and widely known example for it. Deuteromycetes, these are the images. Yes, Pasangla, we are going to conclude. We are going to end our session within 2 slides. Okay. Next, we have to discuss about the kingdom Plantae. Kingdom Plantae. This is also very important. In Kingdom Plantae, can you guys, what are the all members of Kingdom Plantae? Theriyuma? Yarkavadu theriyuma? Please comment in the comment box. Kingdom Plantae includes plants. Enna chodhyam idu ma'am? It includes plants. So plants are the members. Plants are eukaryotic, chlorophyll containing organism with cellulosic cell wall. The cell wall of plants are made up of a substance called cellulose. Some are partially heterotrophic like insectivorous plants are they are heterotrophic. Okay. Yes, plants include algae, bryophyte, pteridophyte, gymnosperms and angiosperm. We will discuss these things in our 3rd chapter, that is plant kingdom. Okay. Yeah, you should remember these points. Plants are eukaryotic. They are eukaryotic, heterotrophic. Some of them are heterotrophic. Most of the plants are autotrophic. Because you know, plants only have the ability to produce what? Only have the ability to produce food in their body. Only have the ability to perform photosynthesis. So they are mainly autotrophs. But some of the plants are what? Heterotrophic. They will feed some insects. Most of the example for it is insectivorous plant. The length of the, we don't have to discuss this thing because we will discuss this in our 3rd chapter. Kingdom Plantae and their reproduction type. We will discuss it in our 3rd chapter. Next one is Kingdom Animalia. Kingdom Animalia, they are multicellular, heterotrophic, eukaryotic organism without a cell wall. A cell wall is absent in Kingdom Animalia. Guys, we are included in this kingdom. We are present in this kingdom. They directly or indirectly depend on plants for food. We are heterotrophic. We need to depend on plants for our food. We have a holozoic mode of nutrition, because holozoic mode of nutrition means it has the ability to consume the food as a whole, and the digestion process will take place inside our body. Okay, and we will show a definite growth. We can only grow till a particular age. Okay, we have neuromotor system mechanism, we have nervous system. Okay, we have the ability to move, we have the ability for locomotion. Okay, and the sexual reproduction is by copulation or the mating of female and male organism, followed by the embryological development. Clear? So this is all about Kingdom Animalia. Pasangala, there are in the first slide, I will show you in the first slide, I already told you about virus, virode and lichen. These 3 are very important, and any of the scientists did not include these 3 in any of this classification. So these 3 are old ones. We have to study about them also. Yeah, so I am going to tell you about virus. We can't put this virus in any of this classification. Virus is a non-cellular organism. It is non-cellular and not true living. This is non-cellular and not true living. Viruses are always known as the biological puzzle. It is biological puzzle because we can't decide whether a virus is living or non-living. So they are not included in 5 kingdom classification. They are not included. Virus is not included in 5 kingdom classification. Virus have an inert crystalline structure outside the living cell. When virus is present outside a living body, it doesn't have any life. It's like an inert crystalline structure. But when it enters into a body, when it comes in contact with a living organism, it will active and it becomes live. Okay, so viruses are obligate parasite. They are complete parasite. They are complete parasite. When they infect a cell, they take over the machinery of that host cell and they will replicate themselves, killing the host completely. That is the process done by virus. Clear? Another doubt? Anybody have it? Any doubt? No. Clear? All good? Yes, we will continue. And virus is a nucleoprotein. It has a protein code. It has a protein code. Virus has a protein code. It is called capsid. It is called capsid. It is called capsid. And it has a genetic material also. Maybe it is RNA or DNA. No virus contain both RNA and DNA. Only one is present. Only either it is RNA or DNA. Okay. The genetic material is infectious. The genetic material of virus is infectious. Generally, virus that infect plants have single-stranded RNA. You should remember these points also. Generally, virus which will infect plants have a single-stranded RNA in it. So, the genetic material of virus which will affect the plants. What is the genetic material of virus which will affect the plants? The single-stranded RNA. Viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. This is also very, very important. Next one, bacteriophage. What do you mean by bacteriophage, Pasangla? What is that? Bacteriophage. The virus that infect bacteria is known as bacteriophage. Some of the virus will infect some of the bacteria. That structure is called or that is called bacteriophage. Usually have double-stranded DNA. The protein called capsid made of small subunit called capsomere. So capsid are made from small subunits that are called that are called capsomere. Capsomeres are arranged in helical or polyhedral. So these capsomeres are arranged in helical or polyhedral geometrical forms. Viruses cause diseases like mumps, smallpox, herpes, influenza and AIDS. These are the diseases caused by virus. Repeat with me guys. What are they? What are the diseases caused by virus? Mumps, smallpox, herpes, influenza and AIDS. In plants, the symptoms can be mosaic formation. In plants, the virus will form mosaic formation, leaf rolling or curling, yellowing or vein clearing, doffing, stunted growth. So, virus is not good for us. Virus can't be considered as living or non-living. When it enters into a living being's body, it can be living or it can be considered as living, or when it goes outside, it is present in an inert crystalline structure. So these scientists, you should know this scientist, Louis Pasteur, D.J. Ivanovsky, M.W. Beijernick and W.M. Stanley. Louis Pasteur, he gave the name virus. And D.J. Ivanovsky, he discovered virus. M.W. Beijernick demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plants and called the fluid as Contagium vivum fluidum. Okay, Contagium vivum fluidum, infectious living fluid. This is the contribution from N.W. Beijernick, and from W.M. Stanley he showed that viruses could be crystallized and crystals consist of largely of proteins. Okay, he made this point. So you should remember the scientist's name also. This is also very, very important. Next, we have to study about viroid and lichen, and we can summarize the session. Now we reach the end of the session. Viroid. Viroid is like a virus. It is an infectious agent with a free low molecular weight RNA and no protein code. Viroid. It is infectious agent. This is also an infectious agent with a free low molecular weight RNA and no protein code. There is no protein code is present in viral. In the case of virus, there is a protein code is present, that is called capsid, right? Yeah. So in the case of viral, there is no protein code. These are smaller than viruses. So viral is smaller than viruses. It is discovered by Theodiner in 1971. It is discovered by Theodiner in 1971. He found that it caused potato spindle tuber disease. He found that it is caused potato spindle tuber disease. Next one is lichen. Lichen is the association, symbiotic association of fungi and algae. Lichen is the combination of what? Fungi and algae. It is a symbiotic. I already told you symbiotic means mutually beneficial association. So the algal component in this lichen is called phycobiont and the fungal component is called mycobiont. Algal component is autotrophic, not heterotrophic, autotrophic, and the fungal component is heterotrophic. So algal component will prepare the food for the fungi. Algae will prepare the food for the fungi, and the fungi provide the shelter and absorb mineral nutrients and water for its partner, for the algae. This is how they are living. Okay, the beneficiary, the mutually beneficial relationship. Lichens are very good pollution indicators. They do not grow in polluted areas. So lichen will not grow in polluted area. So they are the very good indicators, pollution indicators. Clear, Pasangala? Yes. So now we need to summarize the session. In this session, we learned lot and loads of things. Okay, we just discussed about, I will show it. I will show the first mind map so that you can easily connect with the chapter. So in this session, we just discussed about what? We just discussed about, I will use another color. We discussed about 2 kingdom classification, 5 kingdom classification, Monera, Protista, fungi, plantae, and Animalia. Again, Monerans are classified into Archaebacteria, Eubacteria, Mycoplasma. Archaebacteria is of 3 types: halophile, salt-loving; thermoacidophiles, temperature and acid-loving; methanogens, present in gut of some ruminant animals and in marshy areas. Eubacteria is of 2 types: bacteria, cyanobacteria, and then Mycoplasma, the smallest living cell in the world or in the environment. Oh yes. Next, Protista. It is of 5 types: chrysophyte, dinoflagellates, euglenoids, slime mold, and protozoan. Fungi is of 4 types. What are they? Phycomycetes, ascomycetes, basidiomycetes, deuteromycetes. And then plantae and animalia. At the end, we discuss about the viroid, lichen. Is that clear, Pasangala? So, if you like my channel, please comment. So I am winding up this session. Please don't forget to follow my channel for more video like this and like and comment for more videos. Thank you so much, Pasangala. Bye bye. See you in the next class.